Okay, so we've started recording and I'll go ahead and do an introduction and then we'll get started today. So welcome to the ENST seminar. We have a number of guests, which is always very fun to see. We are going to be hearing today from Dietrich Epp Schmidt. So I get the honor of doing seminar, but today also getting to introduce Dietrich. So Dietrich is a PhD candidate who is finishing up in my lab. Dietrich has the distinct honor of being, I think, the longest member of my lab at this point. So he started with me as a master's student a few years back. He came and through a series of kind of dumb luck, I had a project, a new project on urban microbiology, and he became a member of that project and spearheaded an effort with collaborators from all over the world to look at urban soil microbes for the first time. And we had a great time on that project, and he published two really cool papers out of that. And so as he was working and finishing up that master's project, he he was interested in going on for a PhD. I might have nudged him a little bit because I just thought he was all the necessary skills to do that. And I was at the time collaborating with Jude Mall, who's also on today from USDA Beltsville. And Jude and I had a project looking at the effects of glyphosate in glyphosate in Beltsville and in two other locations. And there was a lot of bioinformatics work to be done on that project. It was a huge data set. And to Dietrich's credit, he really has become an expert in bioinformatics, in sequence analysis. He really taught himself just lots and lots of of code and lots of skills that that I'm really impressed with. In fact, I'm going to be he's going to be sorely missed in my lab group because he's really become the resident expert on how to do these types of analyses. Yeah, he's a native Marylander. He's now living in West Virginia, and I think with that, I'll let him get started.
Thank you, Stephanie. So I want to first start by sort of expressing a deep gratitude that I feel towards the ENST community in general, but also towards Stephanie in particular, because I feel like joining the ENST community really was, back in, 2013 at first, was a real changing point in my life, and in terms of having some direction and having some opportunity to work on some projects I was really excited about. It was definitely not a foregone conclusion when I came in. There were some, funding issues early on, and Stephanie was a really great advocate, and the ENST community in general has really been great in terms of helping to support me through my whole grad program. So I feel a lot of gratitude to have had the opportunity to be part of this community for so long. One of the interesting opportunities that has also come up because of my participation with this community has been I've been serving alongside Dr. Kate Tully and Dr. Ray Weil on the Maryland Department of Agriculture Soil Health Advisory Committee. And I got that opportunity in large part thanks to Dr. Weil's advocacy on my behalf with the dean. Basically what this body of, or this group of people do, we're all sort of stakeholders in soil health. Some of us are academics, some of us are members of the government and farmers and representing advocacy groups. And really what we're trying to do is help MDA, Maryland Department of Agriculture, create a framework that will, a policy framework and an incentive framework that will help keep Maryland at the forefront, leading the charge, as it were, in terms of having landscape scale soil health being applied in the agricultural sector. So that's the talk today, I'm going to be focusing a lot on, I'm going to be focusing entirely on soil health. It's a big part of my dissertation is understanding how the microbial community is interpretable in a soil health context. So I think we should probably start off with the definition of soil health. And so NRCS defines soil health as the continued capacity of soil to function as a vital living ecosystem that sustains plants, animals, and humans. This definition is what I'll say is the most recent evolution of our understanding of soils, and it's really intended to draw focus onto soil systems as living, dynamic ecosystems that can be managed, but also that basically provide a lot of the benefits that we want from them, and really to draw focus on the fact that a lot of the benefits that we draw from soils are from the living component of the ecosystem. And so we would be remiss to think of our soils in the agricultural landscape simply as a medium to grow crops without understanding the ways in which the living aspect of the soils, the community that lives within the soils, is providing some of those resources. So there have been a number of ways of measuring these dynamics and these processes in soils. We call these, biological indicators of soil health, and there's quite a few, and they focus on many different aspects of the microbial community and sort of behaviors that the microbial community is engaging in. And so a lot of these are really important, carbon cycling, nitrogen cycling, how much microbial biomass you have, aspects of the microbial biodiversity and their ability to respond to perturbation are really important for understanding how the microbial community and how the ecology of the soil is functioning at a landscape scale. And we've gotten a lot of insights from these different metrics. But taking the perspective of a farmer who's looking for advice on how to manage their microbial ecosystem, many of these indicators struggle to provide concrete managerial or concrete implications for management on the farm scale. So a paper came out just a couple months ago in Soil Biology and Biogeochemistry. It was written by one of the big names in microbial ecology, and basically the paper highlighted the fact that in terms of soil health assessments, microbes and soil microbiology is not very well represented as like a routine soil health as a routine part of the soil health assessment, and that the current metrics in soil health really lack clear interpretability, which is great. And they proposed a number of strategies for dealing with this, and their strategies, I will say, focused a lot on using genetics and genetic sampling of the soils to gain inferences into the microbial communities, which of course that is sort of my area of expertise. So I was really excited about seeing that. But they didn't really provide a clear framework for how to interpret microbial communities based on the DNA analysis that we do. And so there's clearly a lot of opportunity for developing sort of an analysis pipeline that could be usable for farmers as part of a regular soil health assessment. And so this is a large part of what my dissertation focuses around, is about developing the analysis pipeline and sort of a framework for doing an analysis that would be beneficial to farmers, and not just farmers, but really just anyone who has soil that they're looking to manage some sort of ecosystem process or outcome, and being able to have a clear framework for making management decisions. So just to give you a sense of sort of the type of information that we can gain from using genes and genetic analysis in the soils. I'm going to talk about these sort of into into two different categories. We do marker gene surveys, which are primarily what I'm going to talk about today, but then we also have this other set of sampling called that we do metagenomics, and the microbial ecologists, geneticists out there will say metagenomics and RNA-seq are completely different, and I totally agree. They're very different. Lab work goes into them, and you get sort of different sets of information. But I'm lumping them together because in a certain way they provide information about the broader functional diversity of the genomes in the soil, whereas marker gene surveys really focus on a specific gene. And so there's some fundamental differences, and they kind of group together a little bit better. So I'm going to walk through this really quickly. So with a marker gene survey, the information that you get is basically you get an estimation of the diversity and abundance of certain organisms, and or your target organisms, and basically it's kind of like, you know, taking a survey of the forest and getting back the identity of the dominant tree species. Having that information, for example, knowing that your forest is made up primarily of oaks and hickories, tells you something about the ecology, and you're able to infer some functional aspects based on the taxonomy. But it doesn't give you a whole lot of detailed information about what may be happening within that community or the functions of those organisms. You might not be able to assess how stressed the trees are and that sort of thing. And so these types of surveys are pretty low cost, and they capture a wide diversity of organisms. So those are the advantages, whereas sort of the disadvantage is that they have lower specificity in terms of really being able to dive deep into the nuances of the function and sort of what they're capable of doing. In terms of metagenomics and RNA-seq, both of those techniques capture, like metagenomics, you basically sample all the genetic information in the soil, or whereas RNA-seq, what you're doing is you're capturing the pieces of the genetic component that's being actively transcribed. It's actively being used by the organisms to do something. So you're getting sort of a snapshot of what sorts of stresses the organisms are expressing and responding to, what sorts of maintenance activities they're doing, and that sort of thing. So the big advantage here is that you get a lot richer information in terms of the functions that the organisms are engaging in that site at that moment. One of the downsides, though, is that it requires much deeper sequencing and requires a lot more resources in order to actually capture all that information. So a typical sample for a metagenomics data set, you want at least 1 million sequences. Actually, ideally, you probably want more than 5 million sequences. So my shotgun data set that I had, my metagenomics data set that I had in my master's program, had over 1 billion total sequences across all the samples. And this also requires a much more, because there's so much more information, it requires a much more sophisticated bioinformatics pipeline for managing that data into like doing quality control and all that stuff. And then annotating requires a much more sophisticated reference database to get at those functions. So there's advantages and drawbacks. And I'm going to be talking about marker gene surveys today, actually specifically marker gene surveys of fungal communities, primarily because the reference databases and the annotation and quality control pipelines are much closer to being sort of an out-of-the-box sort of service that someone could provide. So in other words, they're much closer to being actually usable at a practical level for someone trying to do large-scale sequence analysis and give recommendations to farmers using just a single sample. And at the end of the talk, I'm going to talk a little bit about the limitations, because it's the, we're getting close, but we're not all the way there yet. So in terms of designing a microbial analysis that really fits the needs of a farmer trying to diagnose issues on their field, I think of a number of criteria that need to be met. First of all, the analysis needs to be able to be contextualized by the objectives of the farmer. So we need to be able to know what their farming system is, and we need to basically be able to address particular desires of the farmer in terms of management outcomes. And so from that end, it really, the analysis needs to focus less on sort of the taxonomy and the what's there aspect, and more on the what are they doing aspect. So the annotation and all of the inferences based off the microbial community really need to be focused on what tangible benefits the farmer is receiving from those organisms. Otherwise, there's no point in trying to manage them if you can't make those inferences. And then of course, it needs to be effective long term as a diagnostic tool, and the management criteria need to lead to tangible outcomes that occur because of management. So there needs to be like this reasonable expectation that if you give a recommendation to a farmer and they engage in management, that over a number of years they should be able to see an outcome in the way that their systems are performing. And then, of course, the analysis itself must be relatively cheap and easy to implement if it's going to be used as a monitoring tool. So it actually, this is one of the reasons, again, that I'm focusing on the marker gene surveys, is because they are actually sufficiently cheap that they could be sort of tacked on the side, potentially, of a typical soil health or soil test. So in terms of setting the standard for this type of interpretation, I want to use what a typical soil test looks like, and sort of the best use of the soil test as sort of the standard for developing a microbial community test, or a soil health test for microbial communities. And so this is a soil test that I just lifted off of the internet. I Googled and I lifted it. And I basically just want to make a few observations. When you do a soil test, basically what you do is you go out to your field, you collect some samples from around your field, you mix them all together, and you send that soil sample out to the lab, and you tell them sort of what you're planning on growing. So they have some sense of what the nutrient demands of your crops are. And so you'll see in this soil test that they have a crop section where they are tailoring the recommendations for a particular set of crops given the results of the soil test that they got. And then you get back like a number of nutrient status type readouts and soil chemistry, and here's pH. And so the way that these soil tests, I mean, there's a temptation to see them as sort of this gold standard of whatever the recommendations in this soil test is, then I have to follow that to the T. But the reality is that there's so much variation in terms of the specific chemistry and dynamics within the soil itself, that these tests are actually best used as sort of long-term management tools, where you incorporate information about how you've been managing your crops, you incorporate information about how your crops have been performing, and then this provides some data to diagnose any issues that you see, or to assess whether you're maintaining the proper balance of nutrients, whether or not you're losing potassium over time or gaining potassium. Is basically tells you whether you should be through time increasing or decreasing your additions. So I'll go on a little tangent in a second here to sort of demonstrate this, but first I wanted to mention quickly to give some background information. So this is my favorite basically plot from soil health and soil fertility. It's a graph of the relative availability of different nutrients according to pH. And you can kind of see that there's this window here of where most nutrients are available. So there's sort of this optimum pH range. Um, and and so that's roughly 6.2, 6.3 to like 7.3 or so. But so my story here is that I have a friend who's starting a farm, and they uh, went out and got a soil test. They sent it to me, and they were like, "What should I be concerned about?" And it looked fine, but their pH was at 6, and they told me that they were working really, really hard to try to get their pH up to 7 or 6.5. Sorry, they were doing a lot of liming and a lot of activities to try to raise the pH. And I asked them whether they'd seen any issues in terms of anything that they'd grown there, having nutrient deficiencies or any problems like that. And they're like, well, no, it's just they were worried about, the long-term uh, productivity of their crops. And what I told them is, well, your crops are the ultimate litmus test. If there's no challenge with your crops, then there's no need to necessarily go out of your way to change the pH. So if they were seeing some sort of nutrient limitation, then they might be able to diagnose it. For example, if they had pH of 5.5 and they were having phosphorus limitation, but their soil test showed that they had lots of phosphorus, then they might want to change their pH. So my point here is that this is the standard that I'm shooting for with the microbial community analysis. I'm not looking for a gold standard where you look at the microbial community and say, oh, this is definitely bad, but more or less looking for ways that you can monitor the microbial community through time and then potentially diagnose problems. Maybe you're seeing disease and you can start to see that disease pop up in your microbial community analysis, and the analysis itself might help you diagnose ways um, of mitigating that problem. So uh, just to sort of recap, in terms of the analysis workflow that I would envision for one of these analyses, is basically you allow the farmer or the producer or whoever is getting the soil test to define what their objectives are. Use those objectives to define the functional groups within the organisms, the sort of guilds or the groups of organisms that you really want to monitor, or that you might want to change their abundance or how they're behaving. And then you do the microbial community analysis. You run it through the pipeline, and you have an output that you can then generate that tells them, gives them some management recommendations. So that's the sort of ideal workflow that we're moving towards. This, of course, begs the question of what are the organisms that are in soils, and how can they be managed for better, better outcomes. And so this is a question that I'm going to dive into in a second. What are the functions that organisms can give us in terms of our soil health? But first, I want to ask you a question, and you don't need to answer. I just want you to think about it. I want you to recognize what your internal bias are. Do we think that fungi are good or bad? And there are a lot of people in the agricultural community that I talk to that have very strong biases one way or another, and it really depends on sort of their personal experiences and where they're coming from. And so just, recognize your the way that you feel about fungi, because we're going to be talking about them a bunch. And to just cut to the punchline of my talk, which is from here on out, which is that different fungi do different things. And so I'm going to quickly run through, I'm going to introduce you to a few of some of my favorite groups of fungi, and then I'm also going to talk about some harmful groups of fungi, just to sort of underline this point that specific groups of fungi do specific things in the microbial community, and that we really need to be aware of these specific functions in order to create recommendations. So the first group that I want to introduce you to is Metarhizium. Some of you may be familiar with this insect. It's the spotted lanternfly, and it's an invasive species that's been wreaking havoc on the orchards, the apple orchards, and the agroforestry systems in Pennsylvania. It's invaded all of the states around Pennsylvania. So we have it in Maryland, in West Virginia, in Virginia, New Jersey, Delaware. All of us now have this spotted lanternfly in some of our counties, and it's been really damaging. But you may notice that this particular organism has little fuzzies around. It's dead, and it's been infected by a fungus in the genus Bavaria, which is one of the genera in Metarhizium. And so the Metarhizium group of fungi are primarily insect pathogens, but they're widespread in soils. They have a diversity of host specificity, so they can attack a wide range of insects, and they're quite adaptable, actually. They're capable of living as insect parasites, but also as facultative saprotrophs. That means that they can, when they need to, live in the soil just decomposing organic matter. And they can also live as plant endophytes. And this is where things get really interesting, because when they're living as plant endophytes, they have been known on occasion to actually provide nitrogen to the plants from insects that they have parasitized. They've been shown to be able to change the way that the plant immune system is activated and change the sort of defensive compound profile of plants and really help plants become more resistant to insect pest pressure. So these are obviously an organism that it would be really great to optimize their function in an agro ecosystem, especially if you're facing pest pressure. But there hasn't really been enough detailed research on them to provide specific recommendations on based on species that you might find in a particular field. Another group that I really love are the Ectomycorrhizae. These are beneficial organisms that form symbiosis with plant roots, and these particular ones have a really specific relationship with certain trees and shrubs, but they typically trade nitrogen also for carbon from the tree. These organisms have faced a lot of interest because in natural ecosystems, they're associated with high rates of carbon sequestration in the soils. And I particularly love them because they're responsible for, they're one of the few microbes that you can harvest biomass from them and sell it at high values. They produce mushrooms, and some of the most expensive mushrooms on the market come from these Ectomycorrhizae. So a couple examples are truffles and matsutake, which are two of basically the most expensive mushrooms on the market, and they're delicious. Another group of root symbionts is the Arbuscular Mycorrhiza. These associate with a much broader group of organisms. About 80% of plants associate with them, and they when you start looking at the literature, they're like a jack of all trades. It's almost as if whatever stress the plant is experiencing, these organisms are under strong selective pressure to try to mitigate that stress. And so they've been really highly associated with phosphorus acquisition, but there's a range of micronutrients and other sort of environmental stressors that have been associated with these organisms. They've also been associated with long-term aggregate stability and good aggregation in soil. So there is certainly, these are one of sort of the darling child fungi of agro ecosystems, as we really want to encourage their abundance. And in case we got too rosy a picture of the microbial community and its benefits, there are a lot of plant pathogens that can really be damaging. Fusarium is one of the most widely known ones. This is a picture of Fusarium wilt in soybeans in Cornell in their experimental plots. But I did want to, I picked Fusarium as a group to talk about in particular because I really want to make this point that you have to have specific information about the organisms that you're sampling, because you can go on the internet and find strains of Fusarium to buy to put on your plants as biocontrol for certain bacterial diseases. And so while Fusarium are largely just thought, you hear Fusarium and think, oh, plant diseases, there are actually specific strains that aren't plant diseases, and things can get muddy pretty quickly. So this is one of the challenges with the microbial community analysis, is that you want to generate enough specific information to actually sort of accurately predict the function of the microbial community. So now that we've met a few of these fungi, the next thing to do is sort of to categorize the types of benefits that a farmer might expect to receive from these organisms. And so I've already talked through a bit some organisms that help with plant nutrition, some organisms that contribute to disease and or disease suppression, organisms that are associated with insect management, soil aggregation, and all of the benefits that come, side benefits that come from that. Some organisms that, the ectomycorrhizae are associated with soil organic carbon, and so on and so forth. Of course, probably most farmers would be most interested in the direct effects that really affect the performance of their systems, but a lot of farmers are also really interested in these environmental co-benefits. And so these are, any of these I think should be on the questionnaire that farmers are looking for to see benefits in their systems. And so we've got a list of fungi, sort of a list of potential functions that these organisms are doing. The next thing is to sort of understand which fungi, like the next step is actually, once we do the analysis and identify these organisms, we have to be able to say something about how agricultural management actually affects them. And so I'm going to spend the rest of my time talking about an experimental system that we sampled from, and sort of analyzing the microbial community within that system, the fungal community within that system, in terms of how these systems are affecting these groups of fungi that we think are important. So which fungi are responding to farming systems, and how are they responding? So the experimental system that we're working in is the Farming Systems Project. It's a USDA initiative that was established back in 1996. And the whole point is to look at different soil health farming systems side by side. So all these strips that you see are different plots and different treatments of farming systems. Basically, within this experiment, there are a number of different farming systems that range from no-till to chisel-till to organic. There's actually several different organic systems, a 2-year rotation, a 3-year rotation, and a 6-year rotation. So this is an ongoing sort of experimental platform, if you will, that is intended to, through time, diagnose and compare the performance of these systems. So we sampled from this system, and in particular, we only looked at 3 out of the farming systems. So we're looking at no-till, chisel-till, and the 3-year organic rotation, because no-till and chisel-till systems were both 3-year rotations. In terms of the crop rotation, these are identical, except that organic has a vetch cover crop instead of a wheat into soy rotation at the end of the no-till and the chisel-till farming systems. In terms of weed control, both weed control and fertility management are the same in no-till and chisel-till, which is that they use herbicides and synthetic NPK fertilizers. The organic system uses tillage as a weed control mechanism, and green manures and animal manures, and then potassium sulfate as the sort of fertility management system. And so those are the broad sort of brushstroke differences between the farming systems that we're going to be looking at. In terms of our experimental design, we took 4 cores, and from these cores, I'm going to be looking at the 0 to AP horizon, primarily because this is where we expect to see the biggest differences among the farming systems. These farming systems were sampled at the wheat phase of rotation in August after the harvest. And so I want to talk a little bit about the soil profile This is Glade. He's the one that did most of the sampling in the field, and I think he's on the call watching this, so I hope I'm not butchering anything that he did. And this is him sort of subdividing the profile into the different subsamples. So um, basically, when you look at a typical soil profile, you have an organic horizon at the top, and immediately below that you have an A horizon. And the A horizon is primarily characterized by sort of organic matter being leached out of the organic horizon above it. But one of the things that happens in agricultural soils where you have tillage is you actually mix these two together. So you sort of destroy your O horizon and you get a distinct what's called plow layer. So this is the AP horizon that is where that O horizon sort of gets mixed in to the A horizon. And so that ends up looking something like, a lighter surface horizon that's deeper. In these farming systems, the sort of rough average depth to the plow layer was about 20 centimeters, but it changed based on where you were in the plot and which farming system you're in. And the sampling scheme that we looked at above the AP horizon is basically 0 to 5 centimeters, 5 centimeters to 10 centimeters, and 10 centimeters to the AP horizon. And so these are, sort of classifications that help us to understand how these fungi are responding, not just across the farming system, but also at a gradient from the surface of the farming system. Okay, so in terms of the soil sampling, we would take soils from each core and depth category and sort of mix them and subsample for DNA extraction. We did the DNA extraction and Illumina sequencing, targeting the ITS region for fungi. So that's how we capture the fungi, and the 16S is how we capture the bacteria and archaea. And then we use quantitative PCR to count the copies of each of these genes. So it helps us estimate the total abundance of the fungal gene copies in the environment and the 16S gene copies in the environment. This is how we get at the fungal to bacterial ratio as well. And then we go through an annotation pipeline of the sequence data where we annotate the taxonomy. And then there's a new fungal traits database that just came out that I'm super excited about. And this fungal traits database is really critical to this analysis because it allows us to assign specific functions to different organisms decomposing different types of different kinds of carbon substrates, but also identifying things like plant pathogens and animal parasites and that sort of thing. And then using both the quantitative information and the sequencing information, we can estimate the abundance of these different fungal groups. And then the final step before doing the linear modeling is to filter the taxa to organisms that are abundant enough or prevalent enough to actually be able to make a really strong inference about the effect of the farming system on those organisms. Because one of the problems with fungal communities in general is they're very patchy, and it's not uncommon to find a fairly abundant organism in one single sample out of the whole experiment, and it's not really useful to say anything about that organism. It's impossible to infer whether that organism is there because of the farming system or it just happened to be there. So we really just sample down to the organisms that we can make strong inferences on. And so then the data analysis is basically, I ran general linear models looking at the total fungal abundance, and then at the different fungal groups aggregated to their functional grouping, and then also individual species to kind of get this layered approach to understand what's happening at the big picture, but also all the way down at the individual species level. And so starting at the top, if we look at the quantitative PCR, we see that that there are both strong depth effects. So we have no-till, chisel-till, and organic organized from surface to depth down to the AP horizon. And we see that there's a strong depth effect of fungal abundance across the farming systems, and there is a significant effect of farming system on the total gene abundance of fungi. So there's roughly 24 million gene copies in both no-till and chisel-till systems, but there's about 32 million copies in the organic system. So that's roughly an 8 million copy increase, or roughly a 30% increase in the total sort of fungal load in the organic system. So to sort of break this down and understand which organisms are responding to the changes in farming system, what I did is I took the specific functional group of classification of the fungi and took and did a linear model of that functional group's abundance due to farming system and sequencing depth. And so this plot is basically, it's a biplot. If you've done microbial ecology, it's essentially the same thing as a differential abundance, except that we're talking about real counts. And the way that you interpret it is that no-till is the reference system, and there's bars for chisel-till and organic. So for example, for the soil saprotrophs, which are just general soil decomposing fungi, or fungi that are decomposing organic matter in soils. The organic, or the chisel-till system, has roughly 20,000 more counts than the no-till, and the organic system has roughly 7,500, 8,000 more counts than the no-till. And so these two bars are directly comparable to each other, and across the different groups, the effect sizes, so in other words, the magnitude and direction of these bars are comparable, but the absolute abundance isn't, because this plot doesn't give you any information about the absolute abundance. And so the point here is to say that there's just a single group, or relatively few groups, that are having a large effect on the total microbial, or are having a large effect that is responding to farming system. And in particular, most of these are actually saprotrophs, so different types of saprotrophs, so different types of decomposers. I do want to point out the Arbuscular Mycorrhizae here. So the Arbuscular Mycorrhizae group is a group that is responding significantly to farming system. And so if we take a deeper look at that, we can see, again, here's no-till, chisel-till, and organic systems. You see that there's an increase in all systems in terms of the Arbuscular Mycorrhizae with depth. And you may remember from earlier, Arbuscular Mycorrhizae are beneficials that are associated with the roots. So it actually makes sense that they're more abundant at 5 to 10 centimeters below the surface than they are right at the surface. But there is a significant decrease in Arbuscular Mycorrhizae across farming systems. In fact, the organic has about an 83% decrease overall in abundance of Arbuscular Mycorrhizae compared to the no-till, and about an 80% compared to chisel-till. So that would be a sort of significant decrease in terms of the abundance of Arbuscular Mycorrhizae in these systems. But we can break it down even further. We can look at individual organisms. And so if we look at the individual species of Arbuscular Mycorrhizae, we see that this effect is really driven by just a few species of Arbuscular Mycorrhizae having a negative association with organic, and that there is actually a species that increases in abundance with organic treatment. And so one of the possibilities that this raises in terms of future research and development is trying to understand what sort of management techniques can exploit this response and potentially offset the overall decrease. And so that's an opportunity that can be sort of pursued in the future. Changing gears a little bit, I wanted to talk a little bit about ectomycorrhizae in this system, because I mentioned them earlier. Ectomycorrhizae have very specific associations, and so we don't actually expect to see any difference in terms of the ectomycorrhizal abundance, and actually there's no significant difference in terms of the ectomycorrhizal abundance. Here, I just wanted to mention it because There's a lot of interest in carbon sequestration in soils, and the ectomycorrhizae are the functional group that is most strongly associated with actually seeing carbon sequestration in soils. And since there's no host plant in these systems that hosts the ectomycorrhizae, most of these, I mean, the gene counts here, the estimated gene counts are from like 20 to 80. These are probably just spores that happen to be in the sample, right? So, so we don't really expect to see that effect or be able to infer sort of a carbon sequestration effect from ectomycorrhizae in these systems. And then another group that is important is the animal parasites. These are the Metarhizium that we talked about. And I just, there's there are some outliers, and this is part of this patchiness, is that I think that in our sampling we actually hit probably an organism that was being parasitized, because there's one sample that's literally off the charts in terms of the abundance of one of these parasites. But overall, there's a decreasing trend between no-till to organic, and it's significant if you exclude this point. So take that however you will. I think the important thing is that I think with these systems, you're going to see sort of a background spore rate, spore abundance, and that and then you are going to have sort of a random chance of really hitting these high load samples. So so that's a thing that makes management and makes monitoring a little bit more complex, but is also sort of this thing where tillage has been associated with higher abundance of these organisms, because it sort of increases the probability that there will be an infection. So it's curious that outside of this one point, that our systems don't seem to follow that trend. But I'm just going to leave that there. I want to transition to making a broader point, which is this is again the same biplot, but looking at all the species that significantly responded to farming system, and they're color coded by their functional groups. And the point that I want to make here is that the majority of the effects and the majority of the large effects are saprotrophs. They're just decomposers. And so when we think back to this, the early analysis with looking at the total fungi abundance, and wondering what was really driving this, it's primarily the abundance of decomposers in the system. And so I want to use this sort of as a springboard to go back to one of these themes of are fungi good and bad? And one of the things I encounter a lot when I'm talking with farmers is that there's this perception that a fungal dominated ecosystem is a healthy ecosystem, and you really want a good fungal to bacterial ratio. And I think this idea comes a lot from these trends that we see in these trends that we see in the natural ecosystems, but it's also it comes out in particular demographics of farmers who are really interested in sort of the ecosystem services and the carbon sequestration. And so these carbon sequestration effects do play out at the biome scale like this, looking at the fungal bacterial ratio. But if you remember from earlier, it's really a small subset of the microbial community that's driving these biome level associations. And those organisms, unless you're working with woody perennials in your farming system, you're not going to be harboring a large component of those particular fungi in your group, in your system. So interpreting the fungal bacterial ratio as a metric to say, yes, my soils are healthy, or no, they're not, is actually really challenging because of this. And I actually jumped ahead of myself a little bit here in the talk, because this is the Nature paper that basically, the biome scale correlations between the fraction of ectomycorrhizal plants in the canopy, or the biomass fraction of ectomycorrhizal plants. Sorry, I misspoke there. And of course, the biomass fraction of ECM plants implies the biomass, implies a higher abundance of ectomycorrhizal fungi in the soils. And it really is specific to this group that's driving those larger trends. And just to sort of try to underscore this point of interpretation in terms of the fungal bacterial ratio, and I'm beating this horse dead because it's one that I encounter so much in the farming community. They're very interested in the fungal bacterial ratios. In no-till systems, we see a very high correlation between the abundance of fungal genes and the abundance of bacterial genes. It's a very neat relationship. But when we start looking at the chisel-till system, we see this relationship starting to break apart, and it really sort of breaks down in the organic system. And the driver between these differences is we see the fungal community increasing in abundance, but we also see like this relationship is driven actually by much larger changes in the bacterial community. And so these two different communities are responding differently to these same inputs. And it's important to understand the nuance of those reactions to really be able to interpret the microbial communities and make an assessment as to how they're going to affect a farmer. And so I would just, caution anyone who's interested in fungal bacterial ratios or any of these big picture metrics to be really careful about what sort of nuances you're glossing over. And so then I just wanted to wrap up the talk really quickly, talking about sort of the database challenges. What I was talking about today are, using taxonomy information to infer function and sort of get at these functionalities. But it requires a whole lot of contextual knowledge and information that needs to also be organized in terms of an analysis that you present to farmers. Needs to be organized in a way that is conducive to a bioinformatics pipeline. And right now, a lot of the databases that we use for bioinformatics are not currently organized to really answer some of these soil health questions, either on the side of identifying the right functional groups to connecting those functional groups to how they respond to specific management decisions. So there's sort of a gap there in terms of going from identifying functional groups to making prescription and making recommendations in terms of the management of the systems. So it's a huge opportunity for development in terms of the databases. We really, there's a lot of information that has been collected that needs to be synthesized and packaged for an analysis pipeline. And there's also a few key knowledge gaps that do need to get filled in. There are also just, unfortunately, a few taxonomy database issues. So I was using taxonomy databases to identify the organisms up front, and one of them is that most of the source material for the taxonomy databases is derived from Europe. So there may be regional challenges getting really fine scale annotation. And then in terms of the bacterial community, which we haven't talked about really today, a lot of the annotations and a lot of the research is dominated by clinical studies. And so there's sort of a bias there. And so there's this opportunity to really develop a soil health oriented reference database, both in terms of the taxonomy and the functions, that has high resolution for identifying specific functional groups of interest. And just to summarize what we've talked about, and I think I'm right on time, really microbiome analysis is on the cusp of being ready for doing routine soil health assessments. There's just a few tweaks, especially for the fungal groups, that to get it there, and there's a little bit more work to be done in the bacterial and the gene annotations to get it to that point. And I'm very skeptical of fungal bacterial ratios. And so with that, I want to thank you for your attention. I want to open up to any discussion points or any questions that you may have. I'd be happy to sort of answer any questions. Thank you.
Thanks very much, Dietrich. Great presentation, and you've already been generating a number of questions that have popped up in the chat box. So maybe we'll try to pick a few of those. Dr. Weil was asking, you made this comparison about Fusarium as being an example of sort of being both beneficial and one that mostly causes problems. Is the same thing true about these, like the Fusarium, with regard to its impact with insects? You know, if it have any negative impacts on organisms like dung beetles or?
Oh, yes, I'm sure actually. Certainly the Metarysium impact organisms that you would consider to be beneficial. That I'm certain that that's true. Yeah. Yeah.
Okay.
So,
all right.
But that I think just to sort of to make the point there though is that sort of what I wanted to get at in terms of this being important as like a diagnostic tool. Say if there's a farmer that's experiencing a real challenge in terms of having an insect pest problem, one of the things that could be on the table is for the farmer to look out for those specific pests being infected by Metarysium and identifying that Metarysium and trying to like develop systems to help get that one Metarysium that's being really successful in infecting the insects more widely distributed in their farming system and sort of help suppress the pests that way. You could do the opposite if you were having beneficials that you were seeing being really highly infected. We could ask the question of what management systems can the farmer put in place to help reduce the pressure on the beneficials by that group. So that's sort of the point I wanted to make.
Good. So there's also a question asking about what kinds of additions that may be coming in sort of part of the treatment. So for example, in your no-till and chisel-till treatments, were the seeds coated with fungicide, you know, the corn and the seeds, and were there additional sort of non-crop? It sounded like there were maybe some non-crop residue carbon components coming in the organic system, like compost and manure, right? So how do you wade through those complications?
I honestly, I don't know that there were any fungicides applied to the seeds. I'm not sure about that. I think that one of the interesting things about this study system is that there are actually different weed management strategies and also different inputs. One of the things that was really interesting is the organic system has significantly higher fungi that are sort of dung decomposing specialists, and it's the one system that has actually like manures applied to it. So we do see that come out a little bit. I just didn't highlight sort of that aspect in this talk. So
okay.
Dietrich, this is Jude from USDA. Yes, fungicides were added to the corn seed and the soybean seed in the conventional systems. The organic systems receive poultry litter, which contains a wood chip based bedding material. So they do get an addition of a composted sort of wood chip based bed material with it. And if you want to look at the mass balances of that, Cavagelli et al. Has published a number of papers on the mass balances of those inputs relative to yield as well as nitrogen inputs.
Also, before I get distracted, I want to just point out, I put back in the chat box a link to an evaluation form, which we would really appreciate you filling out about Dietrich's seminar. It was at the beginning, but it's also just entered back in. So it will only take you a couple of minutes to do, so please do that. So there was a comment, Dietrich, someone suggested consider rendering a microbiota prediction of what might happen if a long-term no-till field was manured and tilled every 5 to 7 years. Is that just as input, I guess? Let's see, do you think there is a danger for farmers from a cost perspective that our ability to characterize the microbial community has surpassed our ability to manage them at the field scale?
Do I think there's a danger for farmers that our ability to characterize the microbial community has surpassed our ability to manage them? at a
So we
field
can,
scale.
we're characterizing the but what can we actually, sort of our level of management versus our level of sort of measuring and documenting these sort of microbial characteristics, I guess.
Yeah.
How do we balance those?
I think that's an issue, certainly. I think that that's sort of where we are right now, is that we have a lot of information that's disaggregated in terms of how management affects different groups that hasn't really been aggregated properly, or in a way that's useful for interpreting. So I think there's a knowledge gap that is definitely sort of ripe for synthesis and ripe for development. I think there are some huge opportunities, especially when you start looking at disease in the fields, and getting specific um, genotype specific information about the pathogens. Really could, if we develop the analytic pipeline, really could provide the basis for choosing which crop varieties you want, and sort of mitigating or anticipating some issues that you might have based off of the particular genotypes of the pathogens that are in your particular soil. So I totally get that question, that right now sort of the place where we're at is we're still not quite getting enough real tangible information for farmers to like make real management decisions on, based on the microbial community. But I do think we're moving into a space where we're getting enough specific information that we will be able to start really making some interesting recommendations in the near future. I'm assuming that we're going to continue doing this work and develop the databases, though, and that's sort of the key to actually being able to generate that sort of recommendation.
And the question that sort of occurs to me, and maybe it's related to this last one a little bit, but you're looking at these farming systems that have sort of been in place for some extended period of time. And so, the idea of sort of change management or adapting management to sort of address, it's like this, seems like it's like this aircraft carrier, that if you're going to turn it and change it, shifting from one management system to another. I mean, maybe there would be, I mean, what do you think? I mean, how dramatically do you think the changes in the microbial communities are going to be reflected in, management systems, in the time frame of a season or two? Do you think, or are we looking at things that are going to take decades to sort of develop?
I think for depending on what the change is, I think that for the most part, a lot of these long-term benefits that we're looking for are sort of things that you accrue over a number of years in consistent management. I also, however, I do think that there are specific management things that people do that can really affect the microbial community pretty rapidly. Although we know what a lot of those are. We know that adding nitrogen and tilling the soil has a pretty dramatic impact on the microbial community. We know that changing, removing trees and perennials from your ecosystem has a pretty drastic effect on the soil and the ecosystem within the soil. So a lot of the things we sort of generally already know. What I'd like to see is a push towards more specific information about specific groups, and particularly like disease causing groups or organisms that help manage disease, because it's one of the places that farmers do spend some money in terms of managing their crops and have some negative environmental side products. You're releasing if you're releasing pesticides into the environment, and you could do it with local genotype fungi, I think that would be a real, benefit.
Yeah.
So,
great. Well,
yeah.
I think we'll probably pull it to a close here, but great seminar. I think very engaging topic, forward